Hello and welcome to this webinar titled HME Lipidic Pellets for Pediatric Application, an investigation of the effect and stability of drug dissolutions, hosted by Biopharma Asia magazine and presented by Dennis Douroumis, Professor in Pharmaceutical Technology and Process Engineering at the University of Greenwich. My name is Stephen Edwards and I will be your moderator. Now please allow me to introduce our presenter today. Dr. Dennis Douroumis is a professor in pharmaceutical technology and process engineering at the University of Kent and Greenwich. His research focuses on the development and characterization of drug products by using continuous manufacturing processes such as hot melt extrusion and spray drying. His research also explores the use of quality by design approaches and process analytical tools for process optimization. He has industrial experience on R&D of solid dosage forms, technology transfer, and scale-up. Due to his expertise, he led several EU-funded projects as a principal investigator, and he has established industrial collaborators for the development of extruded products. He is the head of Medway School of Pharmacy and has joined the editorial boards of more than 10 international peer-reviewed journals, including the Wiley's editorial board for the series in Advances in Pharmaceutical Technology. He has published more than 120 peer-reviewed publications, including 100 full papers, 4 book chapters, and 2 books. I'll now be handing over to our presenter. Welcome, Dr. Douroumis.
Hello, everyone. Today I'm going to present my presentation about hot melt extruded lipidic pellets for pediatric applications, where we actually investigated the effect of the formulation and the stability on the drug dissolution rates. So the outline of my presentation will include an introduction, a brief introduction to pediatric drug delivery, followed by an introduction to hot melt extrusion, and then I will present some cases of drug dissolution and stability of pediatric formulations. So before I start quickly, I would like to let you know that I'm a member of a multidisciplinary group at the University of Greenwich. We are hosted in Medway campus in Kent, which is one of the 3 campuses of the University of Greenwich. And our group works in 3 basic themes. The first one is continuous manufacturing and process analytical tools, where we mainly use hot melt extrusion as a processing technology. We also have a strong interest on medical devices, mainly fabricated by 3D printing approaches. And finally, we also explore nanotechnology, where we develop or engineer a wide range of nanoparticles that can be used for active or passive targeting, mainly for cancer treatment. And without further delay, I would like to start and tell a few words about pediatric oral drug delivery. So the development of an age-appropriate formulation is a challenging task due to a broad range of pharmaceutical and clinical aspects that must be considered in order to ensure the quality, safety, and efficacy of the final product. So most conventional drug delivery systems are not acceptable for pediatric patients, as they differ in their developmental status and dosing requirements from other subsets of population. So, for example, compared to adults or even elderly people. So conventional formulations are not designed for this patient group. It's very common that the manipulation and compounding of adult formulation has become a common practice. And pediatric oral drug delivery formulations or systems include a wide range such as syrups, suspensions, solutions, tablets, capsules, or powders. However, pediatric patients require different oral drug delivery systems than adults due to the difference, as I mentioned, in dosing and administration. So the oral route is generally considered as the most frequent approach and preferred actually to administrate drug doses to children. However, the development of a formulation that can be easily swallowed is a very challenging process. So before we start designing any formulation, there is a range of critical aspects that someone should take into account when working on pediatric formulations. So the very first one is the drug bioavailability. so which is an important aspect, and the fact that the absorption is lower in neonates or infants than older pediatric patients give us a broad range to think about it. So in general, we know that the clearance of drug molecules metabolized in the liver is higher in children compared to adults, and usually they require higher doses per body mass. And also, it's well known that clinical trials in pediatric patients is a challenging process due to different pharmacokinetic needs or variations, which are related mainly to the age of the children, the potentially different doses for different age groups, and also the dose calculated based upon the body mass. So, in concluding, regarding bioavailability, the design of pediatric formulations requires to have high bioavailability, and this is a challenging process. Dose uniformity is also important, as poorly formulated medicines that they don't have a dose uniformity, they usually result in underdosing or overdosing during the course of a treatment. Also, another consideration is the selection of excipients, which is a very important aspect due to their impact on pediatric drug formulations. So most excipients in the formulation in a formulation undergo kinetics similar to the active drug substance, and they may cause severe adverse effects that are not observed in adult populations. So when we run clinical trials with adults, we might not observe any toxicity, but that will have an effect when we administrate the same dose to pediatric populations. Also, many drug substances are extremely bitter or have other undesirable organoleptic properties, which make the development of palatable formulations a difficult challenge. So, for example, the administration of multiparticulates is usually mixed with foods, and it's often indicated, the mixing with food is often indicated to improve the organoleptic properties and thus the acceptability of these formulations. And also, in many cases, dosing aids have been developed, especially for the administration of pediatric medicines. Finally, cultural and societal factors can have a strong effect on children's attitudes and preferences. So, to give an example, there was a market research a few years ago that demonstrated that bubblegum or grape flavors are preferred in the US, where citrus and red berries are preferred in Europe. However, a licorice taste was a preference in Scandinavian countries. So we see that there are a lot of aspects that have to take into account when we formulate pediatric dosage forms. So in the next slide, we see a sort of classification of the various dosage forms and administration routes related to the age of pediatric patients. And if you have a look in the red part of the table, you'll see that we can easily see most of the oral dosage forms are recommended for children above the age of 2, although there's a variety. So the age of children has to be taken into account when we develop these sort of formulations. Also, there's in the next slide we can see a list of excipients which are commonly used for pediatric formulations, and those have an elevated toxicological risk. So the table is a very good example of why special consideration should be given to excipients when designing pediatric formulations. So you see that there are adverse effects for a lot of these materials that are frequently used. So that was a quick introduction on pediatric formulation and what is very important to consider and what we should take in account. And I will move now to extrusion processing. So extrusion process was first patented in 1797 for making lead pipes, and it was just in 1979 where Follonier used the extrusion to manufacture sustained release pellets. So quite similar to what we have done many years later. So over the last 25 years, extrusion process has received enormous interest from pharmaceutical industry in a wide range of applications. So we have the classical hot melt extrusion process, granulation, co-crystallization, and as you can see at the bottom of of the slide, it's obvious that extrusion processing has been known to plastic and food industry for many, many years, and only the last 20 years was adopted from pharmaceutical industry. So, strictly speaking, hot melt extrusion is defined as the process of pumping raw material with a rotating screw under elevated temperature through a die into a product of uniform shape. So what we can see in this slide, we see a schematic diagram of a full extrusion line where we see the extruder and a range of downstream equipment. And from that diagram, we can clearly see that hot melt extrusion processing has a small footprint. So it's an ideal technology for processing. But I'll explain later on for the other reasons that we actually prefer extrusion, or why it is, has attracted a lot of interest from pharmaceutical industry. And why hot melt extruded pellets? There are several reasons why pellets are considered as an excellent option for pediatric medicines, and this is because, first of all, they are easy to produce and manufacture in scalable amounts. So we can see, for example, on the right photo, a full extrusion line which is designed to prepare pellets. So we have the extruder, we feed the material on the top, then the strand that is produced through extrusion, it goes through a conveyor, and then the last bit you see is the pelletizer, where we cut the size of the pellets at the desired size. So we can have sizes of pellets between 1 and 3 millimeters, and then we can collect them straight away. Also, because we can have a precise and controllable size of the pellets, the control of the size is very accurate. We just have to adjust the rotation speed of the knives in the pelletizer. And also another reason is that we can have high drag loadings of extruded formulations. We can see later on in the example that will follow, drag loadings vary from 30, or in some cases we go up to 70%. It's a very important reason why we would prefer extrusion. Also, we can control the drag dissolution rates by adjusting the amount of the drug substance and the materials, in our case the lipids. We'll see also some slides on of how do we control the drug dissolution rates. And also a unique feature of hot melt extrusion is that it can provide or facilitate excellent taste masking properties of the final dosage forms. And as I explained before, palatability is very important for pediatric formulations. There is an option to spheronize the pellets, so instead of having pellets with a cylindrical shape, we can spheronize them and get them in a spherical shape, which is it's an optional process. Also, there's most of the time there's no post-processing of the pellets, so we don't require additional coating of the product that we obtain through hot melt extrusion. And in many cases, finally, they can be used for extemporaneous formulations or other delivery devices. So these are the reasons that we prefer we work on hot melt extruded pellets. And I will start with the first attempt we ever made to produce pediatric formulations. So this is an example where in our first attempt we developed pellets where indomethacin was used as the mold drug substance, and the formulation was designed to co-process a lipid material, in that case was gelucide, also co-processed with a polymer which was a cellulose grade. And this cellulose grade is a pH-dependent polymer. So the aim was not only to increase the solubility of the water-insoluble drug, but also to provide controlled drug release where with 2 hours lag time followed by fast dissolution rates. And also the extrusion process was optimized, and as we can see on the dissolution graph, to obtain pellets with a fast dissolution rate and excellent taste masking, but mainly provide a stable product or stable pellets under stability studies. So in this graph we can see that we actually, by adjusting the formulation and the lipid formulation with the polymer, we achieve to have 2 hours lag time, so there's no drug release at all, And when we change the pH value, we have a very fast release of the drug substance. On the right image, the right photo, you can see pellets. They have a yellowish color and a very uniform particle size of about 1 millimeter. And in this case, the process was continuously monitored by using inline NIR probes, and we analyzed the second derivative of the spectra. So the reason we had the inline monitoring it was to help us to identify the drug excipient interactions by studying the peak shifts of the appearance of nu in the obtained spectra. So I'm not going in much detail, but the analysis demonstrates that the creation of hydrogen bonding between indomethacin and the excipients was the reason that we had very fast dissolution rates, and also we had an effective test masking of the final product. And the next study was involved the development of diclofenac sodium extruded lipid pellets. So the drug substance was co-processed again with another grade of lipid. This time was Compritol 888, an inorganic excipient called fujicalin. So the reason of co-processing was to achieve the desired dissolution rates, provide test masking to the obtained product of the extruded pellets, and improve the stability of the formulations. So in addition, the addition of the organic excipient improved the flowability of the processed powders. And on the right, we can see this time we don't see the pellets, but we see the extruded strands just before we process them through the pelletizer in order to obtain the pellets. And the main goal of this study was to investigate the food effect on the drug dissolution and to what extent the dissolution rates are affected. So for this purpose, we introduced a range of different foods commonly used for administration of pediatric medicines. So, for example, these were the yogurts, applesauce, mashed potatoes, compare them to plain pellets. So we didn't incorporate the pellets into any of these foods. We just had the pellets into the dissolution bath, and also we use a cellulose solution as the control. So all dissolution studies were repeated in various pH values, and as we can see from these two graphs, we studied at pH values of 1.1 and 1.6, and we observed that the release of diclofenac had the same trend. All food grades, initially all food grades resulted in an increase in the dissolution rate, and we can see that applesauce saw the profound effect, followed by yogurt and mashed potatoes. So the main reason for the rapid dissolution rates indeed the fact that there's the presence of high amounts of fiber, which is called pectin. And pectin, there are studies that have shown that it has an effect on the drug dissolution rates. Additionally, another reason was that, and we observed that when we changed the pH values of the dissolution media, it was that the contribution of the pH is very important, because diclofenac is a weak acid, and it can have faster dissolution rates at higher pH values. So we can see that the same effect, the same trend was observed at higher pH values. This time we ran dissolution studies in 5.5 pH. This time we see that we have a very faster dissolution rate, but again the same trend. The food has an effect, and the applesauce has a the greater one, followed by yogurt or mashed potatoes. So the next part of the study was to investigate the stability of the extruded pellets. So we stored them under two different settings. 6 months accelerated conditions at 40 degrees Celsius and 40 and 75% relative humidity, and the long term for 12 months at 25 degrees Celsius at 40% relative humidity. And at the end of each process, the dissolution rates were analyzed again. Unfortunately, we didn't investigate the food effect after stability, but we ran a single pH. And if I remember the top of my head, that was 5.5. So as you can see on the graph, on the right graph, the long-term stability dissolution rates were quite similar to the freshly made pellets, so we don't see any difference. There was a slight increase, which is not significant. However, as you can see, we observe a dramatic change of the dissolution profiles for accelerated conditions, and dissolution rates were significantly changed after 2 hours. Obviously, you can see that under accelerated conditions, the lipidic pellets were not pretty stable. Again, this is not uncommon, and this is probably due to the blooming effect when lipids are stored under higher temperatures. So I remind you that those were stored at 40 degrees for 6 months. So there was a study by Kahn and Craig who report that lipid systems form crystals on the surface and the change in the dissolution behavior are associated with the molecular rearrangements of the lipids. So again, this is not an uncommon and it has been observed by other researchers. So nevertheless, this study showed that the storage conditions should be taken into account and consideration when we work on lipid exudates, and further work might require to optimize the stability of dosage forms. I also forgot to mention that when we collected the samples after 6 months stability studies, we observed that the structure of the pellets had collapsed due to increased temperature and humidity. So this is something we expected. We expect to see a difference on the dissolution profiles. So then we had the following study where we decided this time to investigate the effect of the lipid grades in order to identify lipids that can possibly provide the reproducible dissolution profiles and are not affected during stability studies. So in this study we also investigated the effect of the food grade again on the dissolution rates of the extruded pellets, and by adding the pellets into different food grades. So lipidic pellet composition again mainly of glyceryl bentonate, as was one lipid, and cetostearyl alcohol were extruded with paracetamol as the model API. And as you can see, the loading was very high. We went up to 70% of paracetamol. It was a weight per weight ratio, and just 30% of the lipids. So the two lipids possess different constituents and structures which allow the generation of broad information about the dependency between the observed results and the lipid composition. So in this study, we see thermal analysis and X-ray diffraction graphs of the extruded products. So these were the pellets, but I'm not going into detail. The main outcome of this physicochemical characterization was to observe that paracetamol is mainly crystalline after the extrusion processing. So there was no, it was a very small amount of amorphous material, but the main state of the drug substance was crystalline. And so what happened then was to study the dissolution of paracetamol and the two lipids, and just to inform you that the formulas were designed to provide sustained release of 12 hours, and that's why you see that the prolonged dissolution studies. So the first observation from these two graphs, you can see the two graphs on the slide. The first observation was that the lipids present different dissolution rates. So glyceryl bentonate showed faster, and this is the left dissolution graph. Glyceryl bentonate showed the faster and complete dissolution, complete drug release after 12 hours in food preparations. Again, the food had an effect on the dissolution rate, as in the previous studies we saw an increase of the overall rate. The applesauce preparation showed faster rates, followed by yogurt and mashed potatoes. However, on the right graph, we can see that the cetostearyl alcohol formulations were less affected by the addition of food, and showed very similar results. So, again, on these formulations, the cetostearyl alcohol formulations or pellets, they show an increase of the dissolution rates when we put them in food, but it's quite they're quite similar. So this was a very interesting observation for us, and in the next slide, we investigate the paracetamol release rates under stability, mainly accelerated and long-term conditions according to the ICH guidelines. So for the glycerol behanate, we observed a similar trend as in the previous study, where the long-term conditions, for the long-term conditions, there is no effect on the dissolution rate. We can see that on the left graph, the left dissolution graph, and the dissolution profiles of the long-term and the freshly made pellets are almost identical. However, we can see that under accelerated conditions, the formulation appear quite unstable, and paracetamol dissolution rates increase rapidly. And again, the optical appearance of the pellets prior to dissolution showed the collapse of the structure, so the changes in the dissolution rates were expected. To the contrary, we can see that the cetosteril alcohol formulations show the better resistance under accelerated conditions and dissolution rates applied graph that there is, for the accelerated conditions, there is a slight increase after 8 hours, but again, statistical analysis demonstrate that it is not significant. So we see that the dissolution variation of paracetamol was not that great. And this was very promising for us because we realized that eventually we can identify formulations, lipidic formulations, or actually lipids, where we can control the release profile, but at the same time we can provide sustained release, and the dissolution profiles are not affected under stability studies, although always we have an effect of the food. When we co-administrate these dosage forms with food, we see an increase on the dissolution, but at least they are quite stable under stability. In this last slide, I would like to give you an example, because I mentioned in the very beginning of my presentation that we can adjust. We prefer pellets as a drug delivery system for pediatric formulations, because we can adjust the dissolution rates of the drug substance to as we desire. So in this one, we can see that on the left side, we can see that by adjusting the extruded formulations, so for example, by adding polyvinyl pyrrolidone, which is a well-known polymer, we can facilitate faster dissolution rates according to our specifications. So by adding PVP, we can slow down the release profile, or without PVP, we have faster dissolution rates. So again, by adjusting the amount of the excipient, we can have the right profile we wish. And actually, instead of PVP, we can select a range of pediatric excipients that can be used to, which are safe, they are not toxic, and also will adjust the release profile. On the right graph, we can see two different dissolution profiles of the exact same formulation. The only difference now is that we played with the processing parameters. So we followed two processes. One is known as a cold extrusion. The second one is known as hot extrusion. The cold extrusion is when we process the lipids at least 10 degrees below the melting point of the lipid, and the hot extrusion is when we process the materials close to the melting point or above the melting point. And that was this process was invented by colleagues in Germany. I think it was Professor Breitkreuz. And as you can see, when we process the material through cold extrusion, exactly the same formulation but different temperature profiles, we facilitate a faster dissolution rate. But when we apply hot extrusion, we have slower dissolution rates. And the reason is that during hot extrusion, the drug substance is embedded within the polymeric lipidic material, so we had a control over the dissolution rate. So eventually, in summary, we could say that lipidic pellet formulation can be used for the development of pediatric oral dosage forms. It's a very good drug delivery system, and however, they should be carefully selected for oral administration, as some of the grades are not pretty stable under stability conditions. And also the formulations, lipidic formulations can be optimized, so the effect of food or drug dissolution on the drug dissolution rates is minimized. Eventually, we also saw that all the formulations in the presentation were processed by hot melt extrusion, and eventually extrusion is an excellent technology for the continuous processing of lipidic formulations. So I would like to thank all of you, also our sponsors for our studies, and also all of you for your time and your consideration. I'll be happy to answer all the any questions you might have.
Thank you, Dr. Douroumis, for your great presentation. Now we will begin the question and answer segment of the webinar. But once again, I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now our first question is, what are the main challenges in the development of children's formulation?
There are a few to be honest, there are a few challenges on the development of children formulation. The very first one is that when somebody develops this formulation, it has to take into account, especially in the UK or EU, have to take into account the consider ethical aspects of clinical trials, and it's not easy to get approval for clinical trials for children, and so this is a burden. Then the next issue is that taste masking is very important for pediatric studies, but again, there are ethical restrictions when in order to use pediatric populations, in order to evaluate the taste masking of the formulation. So usually it's very common that adults are used, but adults have a different taste perception compared to children. And also there is a lack of pediatric knowledge, or there's not a lot of information about excipients related to adverse effects when we develop pediatric formulations. I gave an example with the table before, but there are more materials that need further exploitation and further studies to identify whether they could be have toxic effect on children or not. For example, there's a lot of evidence when people use sweeteners or coloring or flavor agents. Some of them could cause carcinogenic results, but there is more to find about excipients. So, briefly, these are the three major challenges when we formulate pediatric formulations.
Thank you. Our next question is, why extemporaneous formulations cannot always be used for pediatric formulations?
Yes, there's a very high risk for such manipulation, because we have a lack of data concerning the stability of these formulations, the dissolution rates, and definitely in many cases there's no indication on the package of the drug product. So in order to have to use extemporaneous formulations, we need the manufacturer to provide information on the packet about the physical, chemical, or even microbiological information of the existing formulation. And usually sophisticated manipulation of adult products involve a change of pharmaceutical form and the dosage, and they should usually be performed by healthcare professionals. So you can go to a pharmacy, you can go to a hospital, and this is also a problem for people for children when they need to administrate the dosage form when they are in a house and they're very sick and there's no possibility to move them. So again, extemporaneous formulation could result to completely different effects from those we anticipate, and definitely they have for sophisticated products. We need assistance from specialized people. So this is one of the reasons why extemporaneous formulations are not always the preferred approach for pediatric formulations.
Thank you. Our next question is, do you consider HME processing as an ideal processing technology for pediatric formulations, and why?
Yes, I believe that hot melt extrusion is an ideal process for pediatric formulations, And the reason is that hot melt extrusion can process a wide range of materials suitable for children. It provides a wide range of dosage forms, so we can have, as we saw today, we can have pellets, but we can also we can easily produce granules with accurate control over the particle size, and subsequently we can use these powders to form tablets or capsules. We can also provide dosage forms with different dissolution rates, and I showed some examples how we can manipulate the dissolution rate of the final dosage form by playing with the by manipulating the processing parameters or even the formulation. And a very important feature of extrusion is that it facilitates excellent test masking of dosage forms. There are several studies where our group, but also other groups in Europe or US showed that extrusion is a unique technology and can help to provide test masking of extremely bitter APIs. And this is a very important aspect when we consider the palatability of pediatric formulations.
Thank you. Our next question is, did you look at two-stage dissolution for EX, starting in acid, then transitioning to pH 5.5?
Yes, we did, but we didn't actually we didn't publish it because we thought that it would be better to provide the dissolution rates at different pH values. We definitely did it, and we can see that there's an increase of the dissolution rate, an additional increase. So as we move, especially with diclofenac sodium, as we move to higher pH values, the release of the drug substance will be much faster. We didn't see that when we had no food at all, so the release was quite stable. But when we use food additives, we saw the boost on the dissolution profile.
And our next question, all these that you mentioned can be achieved by granulation. Then why HME?
Well, if you think about it, granulation, yes, definitely you can do it, but I suspect, at least from my experience, is that hot melt extrusion can provide a better mixing or blending of the API with the drug substance. This is one of the reasons. So it will be easier for us to manipulate the dissolution rates of the drug substance, and it will hot melt extrusion also help us to go to higher drug loading. If you want to, with something again, from my experience, it's not that easy when you go for classical granulation. But if you think also about manufacturing processes, this go a bit beyond the actual formulation of pediatric formulations. Hot melt extrusion it's a technology which can be used in a continuous mode. It is it's highly cost effective. It can take down the cost to 50%, and if you think about manufacturing, and like I said, it's continuous. We don't have to go from batch processing. We in most of the cases I presented, we don't require the addition of, for example, the removal of granulating liquids or a drying step. So it is quite effective. In terms cost effective, and also has lower processing steps compared to classical granulation.
Thank you. Back to two-stage dissolution. Two-stage dissolution to study effect on lipids as well as API?
We didn't do it for the API. We did two-stage dissolution for the lipids. If I get the question right.
And our next question is, are pellets of irregular shape? If yes, coating for taste masking can be an issue.
This is a very good question. I think I mentioned that in my presentation. Usually pellets, if you standardize the process, the pellets have a very accurate size and shape. So if you extrude the material, you obtain these strands. We call them strands. They look like spaghetti. And you adjust the pelletizer so the shape shape is cylindrical with a very accurate size of about 1 millimeter, and you can extend that to 1 to 3 millimeters. So the shape is very irregular. You can only, if you wish, you can change the shape and you can put them in a spheronizer to make spherical pellets. So if somebody wants to have a subsequent coating for taste masking purposes, then this is very possible. The particle size distribution is very uniform. But like I mentioned before, in many cases, not always, but in many cases, the extrusion processing will facilitate taste masking, so an additional coating step would not be required. Of course, that depends on the bitterness of the API.
Thank you for your answers, and thank you, audience members, for your questions. I will conclude the question and answer session. Before I finish the webinar, though, I would like to ask our presenter, Dr. Douroumis, if you have any closing remarks for our audience.
I would like to thank all of them for their time and their attention, and I will be happy if they have further questions. I will be happy to answer if they would like to send them by email to you, and I'll be very pleased to answer any in more detail. Thank you all, and thank also the journal for the magazine for the invitation.
Well, and I would like to take this opportunity to thank our presenter once again. That was Dennis Douroumis, Professor in Pharmaceutical Technology and Process Engineering at University of Greenwich, for sharing his knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send your questions over to me at stephen.edwards@biopharma-asia.com. We'll get those answers back to you. Thank you, everyone. Have a great day.